Hiya. Okay, so this is key area 5 of Unit 1, and it is human genomics. There is only one video to it because it's actually quite a small key area. The amount of stuff you need to know consists of, I think, 3 different definitions and just a general understanding of the idea behind human genomics.
So again, in terms of background knowledge that you have, there's not much in terms of what you would learn in Nat 5 or even Nat 4. There's stuff you probably did in S1 and S2 when you look at DNA and fingerprinting and various forensic things. So you learn obviously the idea DNA is unique to each person. A DNA fingerprint can be produced from a sample of a person's DNA, and that this is responsible for showing us the patterns of the genes in a person. And this can also be used to help not only solve crimes, but also trace genetic diseases and kind of help people know if they potentially have genetic diseases in their own genes or ones they might pass on to their children or ones they might just come across in future life.
Okay, so human genomics, all of the genes in an organism's body are referred to as its genome, okay, not genome. Okay, there are around 19,000 to 20,000 coding genes in the human genome. There's, as we looked at before, there's quite a lot of areas of the genome that don't code for proteins. They don't appear to do anything really. Human genomics is the study of the order of these genes and the base sequences that make up these genes. So that bottom sentence, that's the one. If they ask you what is human genomics, that's the answer to that particular question.
So in Comic Sans.
So yeah, if I know the font is wrong. Okay.
No, it's useful. Okay. It's a bit in a different font because it's a key fact. I just assumed that's what you'd done.
No, I just forgot to change the font. Yeah, the idea is human genomics is a fascinating area of study. The idea is you can look at the order of nucleotides, you can look at base sequences, you can study individual genes, whole sections of genes, and this sort of study, the idea behind it is if we can find out more about our building blocks, we can do much more towards understanding evolution in general. We can understand human disease a lot easier. We can even understand our history, okay? So how we evolved ourselves, even, short-term history up to our 300-year-old history. It can also be used as part of forensics and that sort of thing as well. It's a really interesting study.
Okay, so in terms of sequencing DNA, it's something that's been possible since around 1970. The Sanger method was a popular method, or one of the most popular methods, about working out the order of nucleotides in DNA and RNA, and I believe there is a video link in the SWAY if you were interested in finding out more about this.
It's really clever. So the idea about DNA sequencing, the idea is working out what are the order of the bases. So taking a human person and saying, right, your gene is A, C, 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 C. I'm not going to go for thousands of those bases, but basically working out the order of the bases that should spell out a gene. Now, the Human Genome Project was the first project. It was a global effort in order to map all of the genes in the human genome. It started in 1990. It was completed in 2003. There were a few thousand people working on it all at the same time, and this should give you an idea of how big the human genome was. So the aim was to figure out what are all the genes on a chromosome, or on all of our chromosomes. It's a typical human chromosome. What are the genes, the order of the genes, and what are the order of bases in that genes? So all of the base pairs, that's around 3 billion base pairs in total, were sequenced in every gene. Okay, so that was a large number of bases that they worked out. The order of the base in the hair gene should be this. The order of the bases in the build the cell membrane gene should be this. They still don't know what half the genes do, but they know what some of them do.
Okay, so with respect to the Human Genome Project, there are lots and lots of benefits from it. There are also quite a lot of risks towards it. So in terms of benefits, it gives us more understanding of genetic diseases, which is massively important because it's going to help us in terms of treating them, managing them, predicting them, and just knowing exactly how they work. We're also going to have more knowledge of how the various genes function and what their roles are, and just a general better understanding of evolution and how we became exactly what we are today and how we got there. Although in terms of the risks, it is the exploitation of data to design humans. You know what it's like with data these days. Everything is protected, everything is important. People don't like it being shared. It's a massive data risk, essentially. And also, it can be really, really easy for drug companies to actually exploit this and use it for profit. If we're finding out more about genetic diseases, they can, and then in terms of different medications and things to try and help prevent these, that is massive for drug companies. If they can use this information to make a drug, they can then sell it at ridiculous cost to then make a massive profit from it, which is not really what we want to do.
Yeah, a lot of the problem with this idea, the exploitation as well, is if we start mapping everybody's genome, say everybody gets a DNA test to give you an entire outlay of what genes you have, drug companies, not so much in Britain, but more likely in other countries where they're less regulated,
not free.
they're more likely to start hunting people who have a specific gene, saying, hey, we've got a drug that can help stop you from getting cancer because you've got a particular gene that might give you cancer. Or we've got a drug that's going to help you with your bad back problems because you've got a gene that says you should have a bad back. That kind of targeting from drug companies can be seen as ethically questionable because, hunting for business like that without consultation of a doctor could be seen as a bit iffy. So genomic sequencing allows for the entire nucleotide sequence of a gene to be known, so the order of the bases to be known. Not all people have the same genes, and not all mutations are in the same place in all people. So if we take cystic fibrosis as an example, there's different cystic fibrosis mutations that result in the similar disease characteristics, but they tend to be lighter symptoms or more severe symptoms depending on the mutation. When analyzing a specific gene, computer programs have to be used in order to detect specific sequences, because a human brain would explode trying to look through a sequence of millions of bases and try and spot the odd ones out. A human can't do that quickly enough. It's just not possible. So we use a computer instead, which is going to be much faster and much more accurate than a human person.
Okay, so first of your really important definitions in this key area is about bioinformatics. So bioinformatics is simply the use of computers and statistical analysis of genetic data to compare sequence data. So it's not the nicest definition, if I'm being honest. There's a lot of words to it, but we're about to discuss slightly more exactly what this means. But it's the idea of just really using computers to see patterns and trends in genetic data from people of various populations or the whole world.
Now, the exam type thing that you can get asked about this is it usually asks what are the three aspects of bioinformatics. So it uses computers, statistical analysis, and genetic data. Those are the three things, the triangle that makes up bioinformatics. And yeah, it's a horrible definition, it's a horrible term, but what it studies is fascinating, because what we can do is you could take a whole load of people who've got a particular disease. You can look at their genes and you can say, right, you have all a particular gene in common because we've sequenced your genes against what a normal healthy person should be, and we've identified this particular gene is causing the problem in you. Now, that's the first stages towards helping treat someone. Okay. In addition, what we can also do is we can see trends in common across sort of immigration pathways. So if we look at, for example, Shetland as an example, they've sequenced a lot of DNA in Shetland, and they have found out, to nobody's surprise, that they share a lot of DNA in common with Vikings, as in Scandinavian people right now. So they've got heavily big connections between Scandinavia and Shetland, and that's because Shetland used to belong to, I want to say, Norway, up until, I don't know what year.
The past.
Up until the past, okay? It used to belong to Norway, so, it was mainly populated with, Norwegians. Also, it was, used as a sort of raiding ground. This is why they do the Uphelja festival and that sort of thing, but it's actually in their genes. Most British people show an awful lot of European genes, because the idea is we didn't spring out of the ground in creation times and just be away. We're British. We came from somewhere. We moved throughout the world, and we are descended from common ancestors throughout the world.
Ginger hair is apparently a Viking trait.
Is it?
Mm-hmm. I'm a little bit Viking, hopefully.
Does that mean, like, Ireland, because Ireland's got quite a lot of redheadedness, does that mean that it's Scandinavian links? Okay. So, using genetic data.
Yep, using genetic data. So, some genes have obviously been associated with an increased risk in different diseases, such as Parkinson's, dementia, cancer, and diabetes. There are lots and lots of other ones, but these tend to be the ones that we discuss the most. And actually, that's throughout the whole course, not just this. There is a video here.
I think it's trying to play. I think that's what's going wrong with the slide, is it's trying to play, and for some reason it's also stolen the image from the first slide and the writing from the first slide. I'm hoping it doesn't play.
Yeah.
It's
Skip
trying
it
to
again.
play.
Okay, so second important definition for this is the idea of personalized medicine. And all personalized medicine is, it's the use of genetic data to inform about medical treatment or medical decisions. So it might be a person who has tested to have the BRCA1 gene, which is linked to breast cancer and slightly to ovarian cancer as well, I believe. If someone is tested positive for that, they may decide to have a double mastectomy, and then that massively reduces the chance of them having breast cancer as they get older. So it's taking their genetic information and using it to make informed medical decisions about their own life. So it's personalised, it's about them, it's looking at their genetic information and making a decision that's best for them. And it can also be used to inform what type of drug a person should be issued based on their genome. Is the insulin an example of that?
Kind of. It depends. I don't know what the NHS system around insulin is, if they just give a one-size-fits-all. I do know
in
that
America.
In America, yeah, I do know that certain people suit more one form of or some forms of insulin better than others. But I also know that there's a problem with pricing in America, is that the medical insurance companies will pay for your insulin for you if you've got insurance. And I think the idea is that they will try and go for the cheapest form of insulin every season. So basically it'll be like renewing your broadband or things like that. You'll get a cheaper deal.
And not the one that's more suited to the person.
And that is a great unfairness, because it might mean that a person is taken off an insulin that really suits them and put onto one that really doesn't work as well for them. So they have to use more of it. And if they have to use more of it, they might have to pay for some of it themselves.
Which links to the idea that personalised medicine is so much better if you have money.
Oh yes,
because you can then afford to get the things that are suited to you. If you don't have lots of money, personalised medicine, I wouldn't really bother about it. It's not a thing.
It's even just as simple as like aspirin doesn't work on me, whereas it does work as a perfectly good painkiller on other people. Not every drug's going to work the same on every human, because every human does have different DNA. What we tend to do with the NHS is it's the drug that works on the most number of people with the least bad side effects. So it's kind of it's not great, but
the NHS is great. The NHS is not great. The medicine overall generalness is not.
I will stress that the NHS is the most wonderful thing ever. Yes. But I would say that it is a one-size-fits-all program. It's like school. You go into school, you get an education. They tell you, oh yes, your teachers will look after you and that sort of thing. Realistically, it's a one-size-fits-all thing.
And there are some people that won't always benefit from it, which is where personalised medicine can be useful. It gets around that one-size-fits-all and looks more specifically at a person and their own genetics.
It's an aspiration of what we should be trying to do. We should be trying to do this for everybody, because this is what everybody deserves.
But money.
And people don't deserve money. I don't know. Right, now this is where we get a bit confused. So personalized medicine is using genetic data to make medical choices, such as surgery, or it could be drugs. Pharmacogenetics, this word here, is specifically only about drugs. So it's using genetic information in the choice of drugs. Okay, so personalized medicine is like the big category, and under that you can have pharmacogenetics, where you pick your drug for your genome. And I imagine there's some sort of genetic surgery branch that's surgery based on your genome. Okay, so it's a part of personalized medicine. And again, it's this idea that people respond differently to different drugs. Cancer treatment is a big part of this. So this is something that's offered as part of NHS treatment and that sort of thing. Different people will have different cancer treatments that work better for them and have less terrible side effects. So it might be as part of your cancer treatment that you are put on 3 or 4 different types of drugs to find the one that works best for you. And the aim is to give the right drug to the right person at the right dose with minimum side effects possible. Okay, so those are the aspects of pharmacogenetics, and it is, again, it's an aspirational target that all medical care should be looking for, is to help reduce harm and increase efficacy. And if we can start to do this, if genetic stuff gets cheaper, then what we'll find is overall medicine will get cheaper, because we'll find drugs that work better for individuals, and that means we won't have to go through the cycle of, well, that was terrible, and that drug was terrible, and that drug was terrible. Instead, we can go straight for the drug that's good. We've already covered the insulin thing, but the idea is diabetics often find that one brand of insulin will work better than another. So you can see on these images, it's 2 different brands of insulin that are available. And they do have slightly different characteristics. I know when you did National 5 and you looked at genetic engineering, the idea is you'd produce, this is insulin that's produced via genetic engineering, both of them, but you take a human cell, you take your human insulin gene, and you produce the insulin. But who was the human person that the insulin gene was taken from? Is that person is going to be different to all the diabetics in the world. And the idea is that insulin that the insulin gene that they took from the original human might be slightly different to what that own diabetic person's insulin was like. Okay, so researchers are now using genetic information to predict the most effective type of insulin for a person to reduce the long process of trying to find what fits. A lot of diabetics will have to go through a process of trying an insulin and going, no, that wasn't good for me, and try and find one that actually fits them the best. Whereas if we can use genetic data, we skip that step of having to try what fits, and we should know instantly.
Okay, so ethical considerations, obviously all of these things have massive ethical considerations because it is all not quite playing God, but a bit like playing God. So there's obviously ethical considerations to be taken there. So there are different types of medications that are available. The NHS and the insurance providers, like we've already discussed, they are likely to go for the cheapest one that works for most, that whole one size fits all in general, because it's best for them to take the cheapest option that works the majority of the time. But again, obviously, this isn't the case for everyone. Sometimes that cheapest option might actually be the worst one for them, which is not a good thing. So I really struggle to say this word. Say the word, please.
Pharmacogenetics.
So it's a very costly thing, like all of the personalised medicine. So it may result in higher medical costs if you do decide to go through it. And then that obviously means there's a bit of unfairness in terms of it's only there for the people who have the money. And then it basically says if you've not got enough money, tough, you don't get this kind of nice medicine that might actually work best for you. You just have to put up with whatever you get given, which is not really the most fair situation to be in.
No. Try and say the word.
No.
Even a little bit.
No.
Okay. Right. Okay. So to summarize, for personalized medicine, okay, overall, the idea is, or this is more actually for pharmacogenetics, but the idea is the benefits of it, the right drug is given, but the disadvantages of it could be the individual treatment takes up a lot of doctor time, and doctors cost money, and money is important in life for some reason.
Yeah. Okay. Obviously, a benefit is the right dose is given for that specific person, but again, it's really expensive to design specific treatments. So again, disadvantage, money.
Mm-hmm. A benefit of overall personalized medicine, conditions can be targeted before there are problems. So for example, sending your genetic data off to someone and them telling you might have a risk of cancer or breast cancer, the idea is that you can make a decision about that before it's actually a problem and eliminate your risk or reduce your risks considerably of developing that cancer. But obviously, sorry, I forgot this was me, but obviously that could result in unnecessary treatment. A gene telling you that you're at risk of getting cancer, it's a risk. It's not saying you are definitely going to develop cancer. So say you've got a gene that says you've got a 40% chance of getting cancer and you take massive surgery as a result of that. There's a 60% chance that you might not have had it. And so the person might not actually get the disease or the disorder that the genes are saying they might. And that's unnecessary treatment. That just is. It's unnecessary. It might be good for peace of mind, but in terms of physicalness, it's unnecessary treatment, which is not ideal. Now, that's pretty much it for human genomics. So the idea of bioinformatics, personalized medicine, and pharmacogenetics are really the main core of this, along with the understanding that the human genome is all of the genetic data of a person, and an idea of some of the advantages and disadvantages of the various processes behind it. So it's quite a short key area. In terms of definitions, there's minimal. There's also very few past paper questions about it because it's such a tiny area. If you are a weaker student, so say like you're looking for about a C level pass and that sort of thing, I'd say don't spend massive amounts of time on this key area. I don't think the input is worth the points.
2 marks.
Yeah. It's an interesting area, and if you like it in terms of careers, I actually find it fascinating. If you want to study more about it in your PLP, there's more further reading links. There's so much stuff about genes now because that is the main area of research. But in terms of if you're just looking for these for passing exams, is that this area
one of the most sacrificial areas?
Yes, if you have to sacrifice something, I'd say this is one of the ones. It's interesting and it's nice, and I like it.
Spend more time learning DNA and gene expression than this.
Probably. Next key area is metabolic pathways. That there is going to be heavily asked about. So it's again, it might be a good idea to look through your National 5 stuff about enzymes. Is going to help you a lot with this key area. Okay. Bye.